today I will talk about embryonic organoids morphogenesis, which is a topic I'm working on since around 4 years now, that I came back to France. So this is a topic at the interface between the organoid world and the developmental biology world that are anyway very connected. So I will first briefly introduce the embryonic organoids and gastruloids, which is the object I'm working on field, because I'm not sure everyone knows this field. So briefly, as you know, in developmental biology, now it's very common to study morphogenesis with quantitative tools like fluorescence microscopy. So you have many movies of marine species like zebrafish or insects like Drosophila. But when it comes to mammalian development, there is an obvious obstacle. That is that mammalians are implanted in the uterus, right? So here it's really a basic schematic for physicists like me just to understand mammalian morphogenesis. And you can see the first step. So there is a parallel between human and mouse. And the first step until the implantation are very similar, and they can be studied in the lab. They are studied a lot, actually. And then there is the implantation phase, where actually most of the organogenesis will occur. And then later, you will see the embryo in the yolk sac, in the fluid, and this is what we are used to see with ultrasounds. But the thing is, what is happening in between these two phases is a bit mystery, because you have to dissect in the uterus to extract embryo to see what's happening. So it's very challenging to do live imaging. Some people do for a few days. And obviously for human development, this is just not possible. So over the last years, there have been many efforts to derive what is called embryonic pluripotent stem cells and extraembryonic pluripotent stem cells. So yeah, I don't know if you can see my pointer, but basically at the stage just before implantation, people can extract cells. So these red cells are pluripotent embryonic stem cells. They can give any part of any organ of the embryo. And these blue cells are committed to the placenta development. So people extracted some from mouse or even a human. I'm just going to talk about mouse today. Cells that you can culture in some specific conditions to stay pluripotent. And then you can make organoids with these cells and see what's happening. So there are different combinations, and one that has been studied a lot, and maybe one of the most studied in terms of quantitative way, is the 2D micropattern colonies. So basically, you would have adhesive domains. You would put so these are human embryonic stem cells. So they would just adhere on these patches, and then you would just activate differentiation. But the differentiation factor is everywhere in the factor. But here you can already see just on the bright field image that you have gradients of densities, things. And if you look at the gene expression, actually these patterns, they are able to self-organize with different layers, and these genes correspond to different types of tissues. So brachyury, for example, the red, I'm going to talk a lot about brachyury. It's one of the first genes for mesoderm. So mesoderm is anything that will be muscles, bones, blood. Here, for example, CDX2, it's going to be extraembryonic tissues. And here, SOX2, it's going to be more like neural tissues. So you can see that the system is able to self-organize. And you can see that the size of the pattern is going to influence how it's. So there are typical length scales in this process that are reminiscent of like Turing patterns, for example. So briefly, in morphogenesis, how do cells communicate? There are many ways, but one is like morphogen diffusion. So cells, they can secrete molecules that can be received by other cells, and it's a way to have gradients in tissues and signals. So for example, in an embryo like the Drosophila, one very important morphogen is bicoid. So you can see it in green, and you can see that there is a gradient along the anterior-posterior axis. So here, actually, there is a source term that has been deposited maternally. And actually, if you model just the morphogen dynamics in the tissue, cells can degrade the morphogen, and the morphogen can diffuse. So actually, it's almost like 1D problem, and you will have a diffusion degradation equation for the morphogen that is secreted at the anterior part of the embryo, which explains quite nicely why you have a gradient along a certain length. So in the case of this system, for example, you don't have anything deposited maternally, but you have symmetry breaking already because the cells at the edge are not the same as the one at the center, meaning their morphology is not the same. The cells at the edge of the colony, they have their receptor available to the morphogens that are in the medium, while the cells at the center of the colony, because of tight junctions, these receptors are not accessible. And just this geometrical, this packing thing, is going to create the gradient that leads to that. So there have been other systems that are not 2D, because in 2D the problem is you cannot really make organs, except in specific conditions. So you can also mix embryonic and extra-embryonic stem cells, and then the systems can nicely self-organize in structures that can look like the pre-implantation or post-implantation embryo. So now it's a big field, and people fight a lot to say who's invented the best pseudo-embryo. So we are, in the team, we are working on Gastruloids, which is a bit a simpler system in the sense that we just take embryonic stem cells, but with bigger sizes, and these Gastruloids, actually, they will directly bypass the implantation stages and make parts of organogenesis. And then the question is, what organ do they decide to make? So briefly, the Gastruloids, they have been developed for the last 10 years. And first, they are aggregated in microwells. And what is nice is at the beginning, they are just round and they start to elongate spontaneously. And people have compared the evolution genetically and morphologically with Gastruloids and with mouse embryo. And what you can find, so this is just a PCA for I guess it's single cell sequencing data here, that at the beginning, so you project all the timings on the same reduced space, you can see that at the initial stage, these tissues are very different, but it looks like there is a convergence, and more the tissue self-organize, the more it will be similar to the embryo. So there is something a bit magical of the self-organization, where even if you start with something that did not start at all like the embryo, it kind of recovers the way to form structures like in the embryo. And just so here, at these stages, cells are pluripotent. And here, this is what I'm been focused in the talk. You will start to have this brachyrigine activation, which is a mesoderm. So these cells decide to become like muscles or blood, but they didn't decide yet. What they know is that they are not going to be neurons anymore. And then you have this gradient that is expanded and coupled to a massive elongation that is even further, and you have segments of gene expression. So yeah, in the literature, you have many systems now. It's crazy, and you can see that all these papers are quite recent, actually. So some are more for the pre-gastrulation stages, and some are more for really early organogenesis. And you can see that, I mean, me, when I joined the field, I couldn't understand, because there were all these protocols with mediums. I was like, how do they decide what to put? Because it changes what kind of organ, what kind of part of the embryo you can recapitulate. An intuitive, partial way of looking at that is that in the embryo, actually the embryo is implanted, at least in the mouse, in a specific geometry, so it has mechanical constraints, but also you see the extra-embryonic tissues are polarized compared to the embryonic tissues, and you have many morphogens that are secreted by these tissues. So basically, cells in the embryo, they will receive different concentrations or different kind of morphogens depending on where they are. And my view on these different protocols is that when you put these things or these things, you try to bias the system to be more anterior, posterior, ventral, dorsal. Another interesting thing is people have cultivated Gastruloids in like anti-adhesive wells in like the medium. But what is nice is here, so you have the proper positioning and differentiation of neural and muscular tissues, but only and if only you culture them in a specific range of matrigel concentration, you would get the proper morphogenesis with the shapes. So here it really looks like a neural tube and somites, where here it more looks like a blob. So it looks like the mechanics might play a role, and this is just because I wanted to give a bit of a broad overview before deeping in my small angle of view of the topic. So here, this is human now. This is human, I think, reprogrammed stem cells. And here, it's the first phase when they aggregate, they start to differentiate. So here, you cannot see the genes, but there are many things ongoing. And for the first days, you can see that these organoids, these gastruloids, will start to elongate along the main axis. So already to understand how the tissue does that is quite a challenge. And then in the second phase, they embed them in matrigel on the right or not in matrigel on the left. And you can see the same thing that has been found in mouse, that really you can form somites and massive elongation in the matrigel, where you would not get that here. So of course, matrigel, it's both chemical and mechanical, so we don't really know if it's the stuff in the matrigel or the mechanics of the matrigel, and it's probably a mix of the two, and there are people who are trying to do synthetic gels to uncouple the two aspects. So just to finish on this introduction, so I showed you the mouse things, but now there is a rush to do human structures also. Obviously, there are more ethical problems with humans because normally you cannot culture for more than 14 days. So just to show you that there is a parallel, people work on mouse or human depending on their interests. I guess one of the interests for human is really to use these setups for like screenings of pharmacology and things like that. So okay, first partial conclusion, maybe mechanics plays a role with this story of matrigel. And now I'm going to focus on what I've been investigating when I joined when I created this project and wanted to do first contribution as a biophysicist, because there were many studies on the genetic aspects, but until now there haven't been so many biophysics groups working on this topic. So as I said, many protocols, different structures, you can get gut and hard structures or more like somites, neural tube. But at the end, they all start the same. It's not exactly the same chemicals, but the principle is the same. You take embryonic stem cells, you let them in anti-adhesive wells, because they adhere to each other, they will form an aggregate with an effective surface tension, and then they would round up. And here we have this current pulse, that is the activation, so it's the activation of the differentiation. So it's the equivalent of what I've shown you, what I've been showing you for the 2D patterns, where at some point you trigger the cells to differentiate. If you don't do that, they will differentiate, but in a more variable way. It's interesting, but there is a lot of variability in the organoids field, so it's good that we have at least a way to synchronize them at the beginning. And then what fascinates us is the fact that in just between day 3 to 4, they start to elongate. And that was really the question, why? So in the team, when I joined, so Pierre-François and Ali had already started a bit investigating that at the coarse grain scale. So they were interested in seeing what happens at the genetic level, but also at the more protein level, because a way to make the link between the genetics and the mechanics is to look at proteins like adhesion proteins or cytoskeleton. And so what they had found is between this day 2 and day 3, during the chiron pulse, which triggers really the differentiation. So first, cells here are pluripotent, so they don't express this gene brachyury that I've been talking about. And during the pulse, they would progressively turn on brachyury, not all of them. And in terms of adhesion, there is a very famous adhesion molecule, ikaderine, that is studied a lot in morphogenesis. There are many ikaderines, so if you don't have ikaderine, it doesn't mean that you don't adhere. But this ikaderine is really the most like epithelial-like caderine. And you can see that here, so of course you have growth, but also you start to lose caderine at some parts and keep it at others. And here, between day 3 and 4, you start really to polarize the thing, and you see that at some tip, you have maintained brachyury expression and ikaderine expression, and you lose it at the other side. So the question is, can we study that in a more like cellular detail to try to see how this polarization can be linked to the elongation? So of course you can do like large screening with epifluorescent microscopy. So here, for example, it's the activated gastruloids. Here it's the non-activated ones. So you get many statistics with that. One of the problems is often you don't know, because these are really 3D objects. I forgot to mention, but here the typical size, the typical diameter is 300 microns. And here the problem is sometimes you see 2 spots, 1 spot, but the problem is you don't really know because you don't have the 3D structure, and sometimes they just grow, you know, on the top and then fall down because of gravity. So you can do statistics with that, but it's not really fine-tuned to do a proper mechanical modeling or inference. So me, when I joined, so I had previous experience in 2-photon microscopy, which I love. And these objects actually are very dense to image. I will show you later. So I was lucky that on the IBDM microscopy platform we had a 2-photon that gave very nice results. So this is what I'm going to show most of in the talk, 2-photon data. And here I focus on developing protocols, you know, to study what happens during this stage. So between 3 and 4 days, where really you have the gene expression gradients coupled with the elongation. And here it's inverted microscopy. So you can imagine different ways. So it's not very rapid, right? The order of magnitude is like 1 second to get one plane. Depends how you've been, whatever, but that's the order of magnitude. So you can do in toto imaging, but then it will be at small time scale. So you have to make compromise on phototoxicity, as usual, rapidity. So when I started, I was, because by symmetry, you want to be more at the equatorial plane, because when these objects, they start to elongate, you have more or less a symmetry plane at the equator. So I was imagining more around the equator. So we have cell lines with fluorescent reporters. So for example, this is a fluorescent GFP reporter for brachyury. But what is nice is to have also some mechanical context, some cellular context in parallel. So what I used is also dyes, because when we change cell lines, the problem is they never behave exactly the same, and it's a pain in the field. So me, what I like is work maybe with 6 or 7 well-characterized cell lines, and then if I want to image different objects, I rely a lot on dyes. So here, for example, I use sulforodamine B, which works really well for 2-photon. So you just put it in the medium. It's not toxic if it's at the right dose. It will stand for dead cells, cavity, extracellular space. So you get many informations with that. And just with the 900 nanometer excitation, you get these 2 green and red channels in the NDDs. So here I'm going to show you a typical movie. So it's not a surface, right? It's really deep in the tissue. It's at around like 100 micron, I think, this one. And it's the equatorial plane. So it's like really the equatorial plane. And here you have 2 channels. So each spot that you see is a nucleus. So you can see already that the nuclei are very big in embryonic stem cells. And it's one of our technical problems. So they can occupy like 90% of the cell volume. So which creates like optical problems when you want to segment them, etc. So here, these nuclei, they are green when the gene brachyury is expressed, and they are non-fluorescent when the gene is not expressed. And here you can see the dual image. So here there are holes, but you can see now that there are lumen and cavities. This is a fluorophore. You can see columnar structures here, some dead cells. So I know all of that by experience and analyzing the saturation level. It's not obvious like that. And you can see also gradients of packing here. It looks like you have more space between cells, and here less. So there are many information to look at in these movies. So maybe you can first focus on the right part, which is the genetic part. So this is really the symmetry breaking. So there is salt and paper noise, but you can see that there are zones of negative tissue that appear. And you can see the emergence of the brachyury gradient at the cellular scale. So here it's a 2D image, but it's imaged in 3D, at least around this plane. Usually it's on a 50 micron thick layer. So you have 5 cell layers when you do that. So now I'm just going to play it again. And now you can focus on that. So this is the same thing, but now you can see many things. So these bright spots, I will mention them after. You can see that there are movements, you can see cells that look like almost boiling, and you can see really gradients in texture. So we have many information in these movies. So these bright spots, I didn't know really what it was because I thought it was dead cells, but dead cells usually they are more this size. And actually now in the field it's known that there is a lot of phagocytosis. So the cells that die in this system, they are eaten by the surrounding cells most of the time. And here you can see, so here it's a bit messy, but at the beginning you can really see gradients of packing with here quite packed things and here cells that are more dynamics. So there are many information, and obviously when I joined I was like, yeah, I want to track that. I mean, I knew that it would be difficult, but it's really why you do this kind of movies. So, I mean, disclaimer, we are still not able to fully track, but we work on it. And when you cannot track, you can do many other smart things. You don't need to wait forever to track. So what I did is classical image analysis, like optical flow, it's like PIV. With that, you can really infer the local displacement of the tissue. So it's not at the cell scale, it's more at a mesoscopic scale. So each row here is a group velocity of a cube that is this size. And here, with this technique that is very cheap computationally, you can start to have information that you kind of guess by eye, but that you need to quantify. And here, something that we found is really that, okay, this object is elongating, but the velocity field that leads to the elongation could be in many different ways. There are many internal velocity fields that lead to the same elongation. So having the full map of the velocity field tells you the mechanism of elongation. I mean, doesn't tell you, but gives you clues. And here you can see these red balls of cells. So all these cells flow more or less downward in a block, and you can see a recirculating layer here of cells that flow backward. And this recirculation flow, so okay, I'm going to talk about the recirculation flow just to show you other type of movies. So here the quality is less good, but it's on purpose because I did a multi-parallel imaging with I didn't need a lot of light because here I'm not interested in tracking, I'm interested in doing the optical flow and measuring the gradients. So here it's a SPY 555 DNS stain, which works quite well for optical flow because I wanted to compare that if I measure the velocities on the membranes or on the nuclei, I get the same maps. Another movie, just so here it's interesting because you have the emergence of a gradient, so here you have more cells here. But then what you will see is the gene expression here is sustained, you have a gradient, but then it totally disappears. And it's not in the 3D layer, I checked. So here the cells, they did not maintain the gene expression. What is interesting is here you can see that it did not elongate. So with Simon and Matthias, we teamed up to analyze these flows. So you can do these flows. So here it's a flow, so it's a flow average over 1 hour because there is a lot of noise, of course. Overlapped on the inverted brachyury gradient. And okay, you have different timings, different flows, noise. What can we do with that a bit quantitative? So yeah, partial conclusion. So it's the first time I do partial conclusions, but I think it's maybe good. So collective tissue flows associated with the emergence of a brachyury gradient, it looks like the case. And how do we relate the two? So I'm going to skip that because it's not really interesting. I mean, it's interesting, but it's technical. So just the way we do that is to project these velocity fields in a kind of equivalent of a Fourier base, but in a circular domain. So here we just studied the recirculation flows when the aggregate is not too elongated, close to the circular domain. It's a bit technical to extend that to elliptical domain, and we are not sure we want to do that. So basically, here it's a basis of, so it's like a velocity field basis. So here you have the first radial mode, orthoradial mode, so solid rotation, isotropic growth. Then you have more convergent extension modes and more complicated modes. So this is just a way to project your velocity fields. And here, we did that for many gastroloids, and the nice thing is with this thing, you rescale by the size, so you don't have, because it's not like a Drosophila that is quite reproducible with macrokitty landscapes or things like that. You cannot just average the maps. These gastroloids, they have all shapes a bit different, so you need to have a way to average that, and averaging that in the Fourier, I mean, it's not Fourier, but in this space is a convenient way we found. So then you can find the dominant modes for the gastroloids, and you can see that you have one very dominant mode and then some that are less, and then it shrinks. So then basically we can we decided all the recirculation fields which are experimental and a bit noisy to project them on these first modes that were really dominant in the energy. So if you sum them all you can get a median, you can get a recirculation field, what we call the recirculation mode, and you can do the median or the average between the different fields. So okay, what's the link with the gradient now? Because okay, it looks like the recirculation is oriented with the gradient, but now we can calculate it really. So here the angles are more or less along the line. So then we were like, okay, but what creates this gradient? Is it the flow that creates the gradient? What's happening? So there is a way. I'm not going to go too much in detail because I want to talk about other stuff. But you can basically compute a dipole from the gray levels of your field in your domain, and this is like a vector. And then you can compute the amplitude of this vector along the time. So I forgot to mention, these movies are aligned, which means that the vector anyway is always along the main axis of elongation. And here you can see that the norm of the vector is getting higher, which corresponds to the fact that you have a dipole of gene expression emerging. And then a bit like Johan Smeleish explained yesterday, it's not exactly that, but it's a bit a similar way of seeing things. You can decompose the way this dipole could change. So one way, just cells that decide to differentiate. Like in this half, these cells, they decide to lose the gene expression. It could be also diffusion, it could be more advection. So these cells here, they ballistically move there. There could be division, apoptosis, and all these microscopic components, you can really decompose them in mathematical components that are a bit in the same class. So reaction, diffusion, advection, and growth. And then there is a last term which I'm not going to discuss, which are 3D artifacts. So in this analysis, we can discard movies where the thing is rotating too much in 3D, because here it's a 2D analysis. So if we have this term that is too big, we can just discard the movie because the framework is not adapted. So here it just okay, it's an integral form because it's more robust to noise anyway. So you can compute that because you compute the velocity fields. You have the phi, so phi is a field of gene expression. So you can compute all these things and just have the contribution of these different things. So what we find is during the polarization, initially it's really the reaction diffusion phase that dominates, which means that it's not like you have some gradient that is just amplified by the flow. You have really something active in the cells that mix or change actively their expression. And then later you have the advection that has a higher contribution, and then same thing, you can compare that for different organoids. So the fact that it's reaction diffusion tells you that there is probably some plasticity in the system. It's not like you have a gradient and it's going to be amplified in a kind of rigid way. And a quite intuitive way to see this plasticity is really when you mix two organoids at this stage, they will just re-elongate as one. Yeah, I have 15 minutes left. Okay. So yeah, these experiments really emphasize the fact that the cells, they mix a lot, they can change their fate at this stage. Okay. Then we were like, okay, but how can we relate the flows to the gradient? And what we found is that this gradient is associated to, as we knew that already, to gradients of cadherin expression, and the recirculating population is expressed in snail, which means that there is an epithelial to mesenchymal transition, and that the cells here that recirculate, they kind of become mesenchymal. So you can look at the cadherin the same way on the 2 photons, so it's a bit more tricky because it bleaches a lot, but you can see that there are cadherin islands, cells that lose cadherin, and these cadherin islands, they kind of reorganize to stay at one pole. So if you look a bit intuitively at how really early, you can see these cadherin islands, and when you look at the shear, you can see that they occur at the shear, at the interface between cadherin high and low tissues. So okay, obviously the cells here, they express also adhesion molecules. The tissue is not falling apart. So we did single cell sequencing just to have an objective way because we had too many unknowns on our system. And from this single cell sequencing, we got many things so that we knew already. So one is that the pole that is brachyury positive is also mixed with endoderm cells. So endoderm is all the inner organs like intestines, lung, liver. And you have a kind of cooperation between the endoderm cells and the brachyury that all express ikaderin. So these cells, you see it's a mixed population. It's not like just brachyury cells only, but they do not express exactly the same genes, but what they have in common is really these adhesion proteins. On the overpopulation that is recirculating, we found that it's associated with vascular and cardiac progenitors. So you have a kind of separation between what would be the future endoderm and some mesoderm progenitors, because if you're brachyury, you're a mesoderm progenitor, and more advanced mesoderm cells that migrate back. So we assist to something that is a coupling between the thing that cells take their decisions, but these flows are associated to the fact that they will position in the tissue in something that is maybe meaningful to do organs. So these cells, they express cadherin, and the reverse tissue is expressing osteoblast cadherin. So we found that. So this is cadherin 11. This is a cadherin that is involved a lot in heart morphogenesis, actually. And so if you do the stainings, you have images like that for big aggregates, and it's really reminding of phase separation, where you have islets of icadherin positive cells with less osteoblast cadherin. So here we did the secondary control, and it's not just that it's less packed, but really you have less osteoblast cadherin. So yeah, this is really reminding us of phase separation. So this is classical in a kind of tissue biophysics. If you mix cells which do not have the same adhesion interactions or contractility, if you have enough noise in your system, they would dynamically separate a bit like oil and water. So to recapitulate, we have this system which is a bit like Russian puppets with a mix of pluripotent and endoderm cells, transitioning cells. So here I did the radial stuff, but it's more mixed actually, and cardiac mesoderm. So this is the differentiation level. But if you want to look it in a more simpler way, you could just say that this is a ball at the coarse grain scale of brachyury positive cells, and these things is triggering antagonist gradients of icadherin and osteoblast cadherin. And so now we want to understand why is this thing related to the recirculation. And when we looked at that as physicists, we were like, okay, it really looks like, recirculating flows like Marangoni flows. And for people who are not familiar with Marangoni flows, so actually there are still debates whether the wine tears are really Marangoni flows, but let's assume it is. So here, these are these nice structures that you see in wine. So here, because you have more alcohol evaporation at the meniscus, the alcohol percentage in the water would change the surface tension. And because you have a gradient of surface tension, in physics, if you have a gradient of stress, you would have a dynamics, and here it's a viscous fluid, so you would have a flow. So this is why you would get the wine going up, but then because of gravity at some point it would fall back, so you have a kind of instability. So in our case, can we show more quantitatively that there are gradients of interactions? So one a bit brutal way would be to extract, so these are not cells, these are, if we could extract a tissue that is brachyury and brachyury positive, they could fuse like liquid droplets, and then we could do the same experiments for all these organoids and compare the things. So it's what we did. So just to put a bit of theoretical prior to the experiment. So if you just have two fluids with one liquid-liquid interface and two external surface tension, that can be described by three numbers: this one, and this one. And you can go back to two non-dimensional number. This one is how much these fluids do not like to be in contact. And this one is how much this one has a high surface tension compared to this one. So then you can do an equilibrium phase diagram. And obviously, if this number is very high, it means that whatever the external surface tension, these fluids they do not want to be in contact, so they would stay apart. On the reverse, for example, here you're in a case where the surface tension difference is very high, and then the blue one really doesn't want to have interface with the medium, so it would be embedded in the yellow one. And then you have all intermediate behaviors depending on these two numbers. So in some sense, if you force two aggregates of different nature to fuse, and if you assume that they would reach equilibrium and analyze that, you can kind of infer the kind of relations you have between the surface tension. So this is what I did. So here I took two brachyury organoids and I just incubated them in different cell trackers. So here just epifluorescence microscopy. And you can see that they would migrate. And with this system, you can also start to look at tissue-tissue mixing if you look a bit in 3D. So after a long time, it will start not to be round again. And it's because you have the polarization later and gradients of brachyury appearing. So the question was how to do this experiment, because I don't really have brachyury negative aggregates without dissecting. And when you dissect an organoid, you don't know how much differentiation you could trigger. So what I did is like asymmetric fusion. So I take a whole organoid, which has already the gradient, and small organoids which do not have the gradient. And then, depending on the angle of attack, I can look at how these two tissues merge. So this is like the easiest experiment I've done in this project, and it's quite satisfying because you just put them and they would take different angles, and then you can analyze these angles. So yeah, this is another way to see that, and just to show you, so the plus minus, you can see that they don't fuse. They would stay partially. The angles here are quite high. Well, for example, the plus plus, you can see that they mix a lot. And the hybrid, it's interesting because here, for example, it fills the gradient and it will start to move towards the positive part. So there are signs of surface tension differences, and I will go back to it so we can measure the angles and do the calculation. But then, in terms of modeling, we wanted also to, because what we're interested in the flows, are these Marangoni flows. So we're like, okay, if we take, so this is an experimentally measured field of genetic field, measured coarse grain field. Can we have a theoretical framework that predicts these flows? And so I will be brief because it's a bit technical, but basically we can, it's very common in tissue physics to model tissues as viscous fluids. So this is what we did, it's the simplest thing. But in the equation of Stokes, we can add stresses, and these stresses are interfacial stresses that are related to gradients of cell population. So, okay, no need, I won't have time to discuss that. So this is the easiest term you can write in terms of symmetry. So it makes people laugh usually, but it's true. And this is a term that will convert a gradient of population into an effective surface tension. So here we have parameters. We have same than before. We have the kappas that are how much the low population wants to be in contact with the medium, the high, and how much they want to mix in the ki. So you can do phase diagram of this simulation. And what is nice is that the recirculation emerge quite naturally in a large, broad range of parameters, but they change signs. So here you can see that here it looks more like our recirculation that is here. And yeah, basically I'm not going to go more in detail, but you can do the more quantitative parameter estimation. And what is nice is this parameter estimation is really compatible with our fusion experiments, because you can also, with the same parameter, model the tissue flow and model the fusions. So we think we have at least partially an understanding on why these tissues merge, recirculate and separate. And what is nice in the experiment, if we shake the tissue, so I showed you the fusion experiment, but if we disrupt the tissue, reaggregate it, it will still be possible to form an axis. And actually, when you do the simulation with the range of parameters that we found, So you can see that initially you had a mix of the two populations, so they can separate, and then you will have spontaneously a symmetry break-in. You see that the blue population will start to go at one side, and here actually you would have a recirculation. So okay, so this is our first, let's say, step of our understanding of the. So you know the gastruloid field is huge, and this we did really like physicists like spherical cows or chicken. Like we studied the most basic step we could study, and we are already a bit happy about it. But obviously there are many things to look at. So here. I just described the early flows of the early symmetry break-in, but when you look at these movies, so here it's described by the Marangoni, but then what we didn't study is the main axis elongation, and here we do not think it's only due to Marangoni flows. We think they are active stress because the cell layers at the edges, they can exert contractile force, so this is something we are studying now. And I have only like 4 minutes left, and I was half of my presentation, so I will just give you an overview of the other things we do in the lab, and I would be happy to discuss about it if you want. So, something I told you that these are cardiac progenitors that recirculate this population, and we started playing a bit. Okay, I started playing a bit and culturing a bit longer in different protocols. And so this is where we were, and I started playing with protocols, and then actually if you could let them evolve a bit more, so here it's not organogenesis, right? It's just patterning and positioning. But one more day after, you can really see, so this tissue starts to make a vascular like network. So this is a vascularization marker with some cavities, and here you have tubes and more like epithelial or endothelial like tissues. So you can study that dynamically with 2 photons. So here it's quite big, but I use like air objective and low resolution, because here I really want to look at how these cavities evolved. So this is an overnight movie, and you can do that on many because I don't sample a lot on time. And you can see this is a big tube that is remodeling here. So these are different Z-planes. And here on the top you can see the emergence of the cavity. So here this is the endoderm. So these tissues are more like gut tissues. And here, these tissues are more like partially cardiac tissues. So you can see the emergence in the movie of this first. So I wouldn't say cardiac pocket because I cannot say that. I don't have the staining for that. But these pockets here, few days after, when we image, they are beaten. So now this is time averaging, image on time. And you can see the tissue pulse, so it's scanning, so you have a bit of interplay between the scanning and the beating, but you can really see the beating tissues. Here, also, this is a zoom now, the organoids are much bigger, and these are vascular cells that start to emerge, and here these cells express like CTNT, which are cardiac marker. So this is really something interesting that we start to explore with collaborators in Latimon, because we are not experts in cardiac development, but this is really exciting also in terms of deep imaging, because now the organoids are more 1 millimeter big, and yeah, if we could do like a 3 photon or photoacoustic imaging, that would be great, and this Mifobio meeting was great to get these ideas. So it's a pity, but I didn't have time really to talk about all the quantitative efforts that we do in the team to push to the cellular scale. So we have Alice, who's a PhD in the team. So she's doing a lot of efforts to quantify division live and fixed. So just to show you, so a division live, so you can see that the cells are very dense, but a division even live, you can see it quite easily. So it's a typical task for deep learning. And we have been collaborating with so yeah, so then you need to align in 3D. So we work a lot. Alice is co-supervised by Leo also on how you register in 3D movies. And we have been collaborating with Jean-François Ruprecht and engineers from the INRIA to just try a 3D UNet plus time network, where the network would see not only the time of the division. So what really matters for us is this dipole, right? But what you give the network is a 3D image. So here it's 2D, but we give a cube of these 3 consecutive times. So Alice has annotated many divisions. and then, so I cannot give you. I mean, the big movies where you can really see the divisions on top of the detection was crashing on my computer. So this is very preliminary, but these are the first results of the network. So here, these are like a probability of having a division, and this is the inferred orientation. And actually, qualitatively, it's quite good. I mean, we have at least 75% of like things that are really like the division at the right timing. And now we need to explore the data to see what are the false positive, but it looks very promising. So we do a lot of fixed imaging with transparization. So here it's a Z-stack on 2 photons with the division marker fixed. So Alice is really looking at are the divisions randomly oriented in the tissue, things like that. So for big aggregates, you can do maps like that. So here each point is a division. It's very easy to segment here. And then we can do coarse-grained maps of division within the Gastruloids. So there are big gradients of division probabilities. But then one difficulty is there are also big gradients of cell packing. So here you have a high node of division, but it's maybe just because there are more cells, the cells are more packed. And here we come back to this challenge of segmenting the cells, which is very challenging in this system because you can see that the nuclei look a bit ugly. And I remind you that this is 2-photon imaging, and we want to do it also for the live. So yeah, basically we have developed adapted protocols to do like immunostaining with 4 color on the 2-photon. So I'm done. And yeah, basically what we do is we call that the light sheet for the poor. So we embed our many organoids in a double cover slip setup. And then we image all organoids on one side on the 2-photon, then flip, and then merge the sides. So it enables us to get the 2 sides fused, which doubles the deepness at which we can go. So yeah, I'm going to conclude. So yeah, we work a lot on STARDIS segmentation. So we have really difficulties to validate, because sometimes STARDIS predicts things and maybe STARDIS is more right than us. So we are really in this process of trying to find ways to validate. So our students, they developed also an APARI pipeline to visualize density maps with the STARDIS and co-localize them with gene expression maps. But obviously, when you do 3D, you have a shadows effect, and we are really brainstorming on how to correct this shadows effect, because you can see gradients, but it's just the gradients due to the shape of the organoid. And to conclude, we have also Jules, who's doing many efforts on tracking. So tracking on sparse data is okay. So here it's a mosaic, so half of cells are only fluorescent. But the problem is in organoids, mosaics create many problems, because it means usually you mix 2 lines, and these lines, they can not exactly have the same behavior, and you might observe artifactual things. So we really want to do the tracking in a dense environment. And for that, we have also Valentin, who's doing SOSPIM live imaging. I think he will be very happy joining Mifobio in 2 years. And now, yeah, so we put our efforts in more like, so we do like simpler experiments, because imagine the recirculation, you need to be at the proper timing. But now we do really design experiment where we explore the imaging parameters just for the tracking problem. So I do a lot of fusion experiments for Jules, because it's a nice physical problem that is quite determined. You know, these aggregates fuse because there is surface tension, so the flow is quite deterministic. And the challenge for Jules is really to segment these things and track them, taking use also of the division detections. So, okay, thank you so much for your... so I had all a part that I couldn't talk about microfluidics, organoid in microfluidics and 2-photons. So if you're interested, I would be happy discussing. And I thank all the people involved here. I'm very grateful since I joined the IBDM. It's a great environment, great students. So I'm very grateful, and thank you for your attention.
Thank you, Sham. Thank you very much for that great presentation. One question was, do you have an idea of who's coming first? Is it the BRA activation or the change in ECAD expression?
So this is what we are looking at now. And it's clearly the BRA activation, because the BRA Curie cells, they have different levels. They are first ecadherin positive, and then if they transition further, they can lose ecadherin. but they can also, we think, maintain in the ikaderin positive state, which is not pure ikaderin, it's also N-caderin. So it's clearly BRA that comes first.
And tiny question or point, when you say that you cannot do mixed population to have fluorescent, could you use the same population, but half of it you just inject a dye, and in the other population you don't, and like this they have the same biological properties, but just dye or no dye?
So yeah, we've played a lot with that because we're really hopeful for that. The problem is these cells, they divide every 6 hours. So you have a tremendous dilution of the signal. And the problem is the processes that we look at, once you mix, the mixing part is artifactual. So you need to wait that it equilibrates again. And until now, we don't have really good results, unfortunately, with that. But yeah, that's really the hope. So now what we are doing is just our own cell lines, but derived from the same line with different histone fluorophore. Fluorescent.
Any other question? No, okay. So with that, we thanks. Oh, okay, sorry.
A small technical question about how many days can your gastruloids live under the microscope? Do you need to refresh the medium?
So that's a good question. So under the microscope, to be honest, I never do more than one consecutive day, because it's never like an incubator. So it can live, but it doesn't look like after 3 days in the microscope incubator, it won't look exactly as the one in the incubator. So you can not change the medium for many days. It would differentiate in stuff, but yeah, the thing is at some point you need to have a protocol for so they don't die. I mean, there is partial death, but you can leave them without changing the medium for 2 months, and there are still parts of the tissue that develop that do organs that I have no clue. I guess the medium is very acid, so I don't know what it's doing. But yeah, for the hard thing that we are trying to push now, usually I don't change the medium, but I just we just every day, you know, put because we have a lot of medium actually in the 96 well, but we just make a flush, you know, to re-equilibrate, kick dead cells, and but yeah, most of protocols, they start to shake after a while, but if we can avoid that, yeah. So depends on what protocol you want to do and what organ you want to do.
Okay, thank you.